Dear students, today we'll discuss the anatomy of monocot root. In the previous lecture, we discussed the anatomy of dicot root, and today we'll see that monocot root exhibits some similarities with the dicot root as well as some dissimilarities, and we will take those similarities and dissimilarities one by one. Now, as in case of the dicot root, the outermost layer in case of the monocot root as well is the epibilema or the epidermis. So the outer most layer of monocot root is also epidermis. It consists of the barrel-shaped cells, and some of these cells give rise to the unicellular root hairs which help in the absorption of the water. And then inside the epidermis you have the parenchymates cortex. You have the parenchymates cortex. This cortex is made up of the parenchymates cells. This is the second similarity. You have the epidermis. It is also present in the dicot roots and also in case of the monocot roots, and some of the cells of the epidermis give rise to unicellular root hairs. Third is endodermis, and this endodermis is the innermost layer of the cortex, and the cells of the endodermis, they contain suberin coating. They contain suberin coating on their radial walls. They contain a coating on their radial walls which is known as Casparian strip. This we have already uh, discussed in the previous lecture. Casparian strip, and this Casparian strip, it is made up of water resistant or water impermeable substance, that is suberin, and some of the cells lack this suberin deposition, and those cells allow the movement of the water from the cortex into the vascular tissues. Then that tissue is, and those cells are known as passage cells, and some of the cells in the endodermis, they lack this Casparian strip. Such cells are known as passage cells. So these are all similar. These characters are also present in case of the dicot root. So until now we haven't seen any difference between the monocot and the dicot root. So from when we move inside, then we have pericycle. Pericycle in this case gives rise to the lateral roots. In case of dicots, we observed that in this pericycle, it has two functions. Pericycle has two functions in case of the dicot root. First is that it forms the cambium ring. It forms the cambium ring and helps in secondary growth. And second, it forms the lateral roots. It forms the lateral roots. So this pericycle in case of the dicot roots has two functions: the formation of the cambium ring and the lateral roots, or the secondary growth and the lateral roots, while as the secondary growth does not take place in case of the monocot root. So the only function in case of the monocot roots that pericycle has is the development of the lateral roots. So the function of pericycle in case of monocots is only the formation or development of lateral roots. So it helps in the formation or the development of lateral roots, whereas in case of the dicot roots, it has two functions. It has function in secondary growth, and it has a role in the development of the lateral roots. Now this is one. Second, there is no secondary growth. No secondary growth. You will not see any secondary growth in the monocot root under normal condition. No secondary growth. Third important point is that in case of the dicot root, the number of vascular bundles was less than 6. It was less than 6, and generally it was 4. But here, in case of the monocot root, you will see the number of vascular bundles, the number of xylem bundles or the phloem bundle is more than 6. It is always more than 6. More than 6. And in case of dicots, generally the number of xylem and phloem bundles was 4, and at that time it was known as tetrarch condition. So here it's more than 6, it's known as polyarch condition. Polyarch. So you have more than 6, for example 1, 2, 3, 4, 5, 6, 7 and 8. Here we have 8 xylem strands and 8 phloem strands. So the condition is polyarch condition because we have more than 6. In case of dicot root, the most prevalent condition is the tetrarch condition, when you have 4 xylem and 4 phloem, these bundles. Then the condition is radial, the arrangement is radial, so the xylem strands and the phloem strands, they are radially arranged. They are arranged radially. You have separate xylem and phloem strands, unlike the in case of stem. Now another point is that the condition is exarch. The condition is exarch. The condition of the xylem is exarch. You have the protoxylem towards the outer side and the metaxylem towards the inner side. You have protoxylem towards the outer side and metaxylem towards the inner side. This condition is known as exarch. So radial arrangement and exarch condition, it was also found in dicot. So it's not difference, it is the similarity. And another important point is that the cells in between these vascular bundles form the conjunctive tissue, and the conjunctive tissue in case of dicots was parenchymates, but here in case of monocots it may be sclerenchymates, and the center of a monocot root is occupied by a large section of parenchymate cells, and it's known as pith. And this pith is well developed in case of monocot root, while it is less developed in case of the dicot root. So we'll again have a look at the anatomy of the monocot root, just to remember the main points. So the outermost layer in case of the monocot root is the epidermis, and some of the epidermal cells form elongated unicellular root hairs which help in the absorption of the water, and this is followed by a multi-layered cortex consisting of the parenchymate cells. Now the innermost layer of this parenchymates cortex is the endodermis, which contains the barrel-shaped cells having suberin depositions, and which is known as the Casparian strip. Some of the cells lack this Casparian strip, which are known as passage cells. Then inner to this is the pericycle. Pericycle has the function of development of the lateral roots. Then inside this you have the polyauric condition of the vascular bundles. The vascular bundles are more than 6, which is the polyauric condition, and then they have a radial arrangement. The xylem has exarch condition, protoxylem facing toward the outer side and metaxylem facing toward the inner side, and then you have the pith, which is highly developed in case of monocot root, and there is no secondary growth in case of the monocot root. Thank you very much.